afternoon. I come to you from the University of Dallas, where I've been working in conjunction with several people on investigating coral growth at the organismal and molecular levels. So I have to start this talk with describing how the project began. So I visited Moat Marine Laboratory in February of this year, and I met Dave, and he described to me all of the different things that go into microfragmentation. And I asked him, as I like to ask most of my biologist friends, what are your biggest unanswered questions? And one of those questions that he said was, well, we know that when we microfragment, that there's exponential tissue growth, and it seems to grow a lot faster than in the larger pieces that we used to do fragmenting with. And so he said, I'm just wondering what genes and what biochemical pathways are involved with that, like what's going on. And so I went back to the University of Dallas, and I talked to the geneticist that we have on staff, and he and I, as we're talking, he said, well, what parts of that microfragment undergo the most rapid growth? And how fast does that tissue grow? And so I said, I don't know. So I actually asked Dave, like, do you have any data on this? And he said, he sent me one little video clip. And I said, well, let's start to collect some data on this. So this really was the first step in this project. So this project was completed with the help of John Paul Diffenthaler and Mason Cosgrove, who are two students at the University of Dallas. And we started, actually JP started over the summer when we got the microfragments, trying to figure out how are we going to actually take these pictures. So we have them in the microfragments in a tank. We were taking pictures in the tank, but then I also have this microscope that I actually use for other work. And I said, well, we should try to see if we could get more close-up pictures using this scope. And so what we did was we took a number of different types of shots using this microscope setup, and we landed on one particular shot that really worked for us. At first we were taking them at all different levels and the edge, and so JP did an excellent job really hammering out a protocol. So we have microfragments of Orbicella flaviolata that came to us. We took daily pictures, which ultimately were created into a time-lapse video, and that's what Mason did. She was responsible for taking all the pictures that JP had taken and putting them into a time-lapse video. Then we did image analysis using ImageJ, and we collected a number of different data points, and I'll show you some, data related to polyp formation. So one thing though, before I show you the data, is I wanted to show you the time-lapse video. And I want you to really watch the edge. So here, watch that up here. This is over the time course of 11 weeks after we got the corals in. And it does jump around a little bit. We've learned some things since then, so when we undergo this project to increase our sample size, hopefully, we'll do a little bit better than we did at the first time. So one thing that we were concerned with is by taking the corals out of their main tank and putting them underneath with a microscope, how much stress were they going to be under? So we limited our sample size to just a couple of fragments because I didn't want to stress them out too much. But one thing that I did want to go back to this video and mention is I want you to actually look at this particular polyp that I have in red, circled in red. Over the time course of just about a month, you can see that the polyp has increased in size quite a bit. And on top of it, in this picture from August, this first picture on the left here is from July 23rd of this summer, and the one on the right is from August. And you can actually see the beginnings of a baby tiny coral polyp. And so if we keep a hold of that, the thing that was most amazing to me is that even over the time course of the next 11 days, you can visualize growth, and you can see those tentacles that were starting to form. So to look at this more graphically, over the first 4 weeks that they were in lab, this is the center polyps, this is the size of the center polyps in centimeters, and then this is the baby polyps. And we took from the very first time we could see the polyps visually, measured them, and took that as week 1. So over the first 4 weeks of their life, they increased to nearly 50% of the centers. Also, the other interesting thing is we only saw polyp formation on the edge. So there was no new polyps that were formed in the center. So this information was critical to us to be able to begin to answer the second question. We needed to know, in order to genetically analyze rapid tissue growth, we needed to know which tissues were growing the fastest. So to go on to the second research question, we wanted to know if the HIPPO growth pathway, which I'll talk about in just a second, was involved with edge tissue expansion. And again, JP helped me with primer development for this project. The other interesting personal note is that when I was at Moat in February, I met Hayden Tompkins. And as you can see, he was wearing a Texas Rangers baseball hat. And I said, Hayden, are you from Texas? He said, yeah, I'm from Dallas. I said, no way. And so I said, when you're done with your internship at Moat, what are you doing? And he said, well, he's like, I don't know, I might come back to Dallas. I said, if you do, let me know. I was like, I can always put you on as a visiting researcher in my lab. So ultimately, that's actually what happened. So Hayden came to me he was doing, actually the day that I met him, he was physically making the plugs. But he ultimately went on to also do a project in ocean acidification at Moat, and that project ended in September for him, and so he came back to Dallas and has been working in my lab this past semester. So the HIPPO growth pathway is a pathway that is fairly complex, so I'm just going to focus, though, here on this protein down here called cyclin E. So cyclin E is a protein that, when expressed, it actually initiates the S phase of mitosis, which is cellular division. And so when we have growth happening, cyclin E is present. And when no growth is happening, cyclin E is absent. So what JP did was he developed primers, actually, that were obtained from a sequence from Ileana Baum's lab at Penn State. And we developed primers to examine cyclin E. So there was 3 different primers that we used. And so what I did was we removed tissue from the edge and from the center of the microfragments. And you can see that here. This is prior to tissue removal. And then I took tissue from the center and I took tissue from the edge. And Hayden helped me to isolate the RNA, go through PCR, cDNA creation, and then use those HIPPO growth rate primers that JP actually developed in order to examine the tissue in those regions that I had isolated. So the other interesting side note here is that, and I was a little bit shocked, when I was literally dissecting, it was almost difficult for me emotionally, which is weird, but I was like, I'm killing the tissue, and it was sad. But I came back to those same samples 2 weeks later. Look at this. You can't tell. You can't even tell where I took that from. So it was right here and right in through here. And I was kind of shocked. So that actually has spawn a new research idea for me. So I'm actually JP is presenting a poster later tonight, so if you are very interested in the genetic analysis and the more nitty-gritty details of that genetic analysis, you should see him tonight. So I'm just going to give you a little insight into these results. So we are looking at RT-qPCR, and so we're looking at relative expression of the cyclin E protein and across 3 different primers. So the different primers basically look at different portions of that cyclin E gene. And so the good thing is they're all telling us the same thing, which is verification. But the weird thing to me and to my geneticist was that the center actually has greater expression of cyclin E than the edge, which was inverse of what we were predicting going in. Although since I've been here, I've talked to a few people and I have some ideas about why that actually might be. So I might look into some of those in the future. So the conclusions of this particular study are that number one, I wanted to know what parts of the microfragment undergo the most rapid growth. And what I found is that the edge is really where rapid tissue expansion is happening and new polyp formation is happening. And that it took approximately 4 weeks for the edge polyps to grow to about 50% the size of the center polyps. So that is very rapid. And we saw all new polyps that were actually around the edge as well. And then finally, I wanted to know if the HIPPO growth pathway could be involved in the edge tissue expansion. Now, one thing I didn't mention about the HIPPO growth pathway is that it's been previously discovered in Drosophila and in humans, but it had never been investigated, to my knowledge, it's never been investigated in coral. So the one thing that we did find is that the HIPPO growth pathway and cyclin E is actually present in Orbicella flaviolata. And so that is something that we definitely know is present and is operating, even if it's not operating the way that we predicted. So the future directions are that we are going to continue to investigate the HIPPO growth pathway. We really need to increase our sample size. Some of our error bars are large because our sample size was low. I wanted to be conservative initially so that I could make sure that I maintain my samples. So as a result, I really need to get more samples in order to increase my number of replicates, both for the growth study as well as the genetic analysis. But the other interesting part of this was the recovery from injury. So basically, I injured these microfragments, and that recovery from that injury was quite rapid. And so the other thing that I would like to do is then go back in and after injury actually analyze gene expression while the recovery from that injury is happening. So that's a new future direction that I would like to go. Finally, though, I really have to thank my team. So Dr. Drew Stennison is our geneticist, and then JP and Mason are my undergraduate researchers. Of course, I have to thank Dave Vaughn and Hayden Tompkins, who both, without Mo, this whole project would have never happened. But actually, ultimately, I have to say that when I went in February, I was going to visit my aunt and uncle. My aunt and uncle have lived in the Lower Keys for over 30 years, and my uncle happens to be Bill Becker. If you're in the Lower Key area, he's the US 1 news radio guy, has been for many years, but before that, he actually has his master's in marine biology, and he ran Sea Camp off of Big Pine for many, many years. And so I was teaching a new marine biology class because although my area of expertise is in the evolution of reproduction, and most of my dissertation and my postdoc work has been in a freshwater snail from New Zealand, my love has always been the sea and marine life, and I've taken students to Marine Lab for many years and been diving since 2000. So I was telling him I'm teaching this marine biology class, and he said, hey, you're coming down in February? I should introduce you to my good friend David Vaughn. And so that I have to thank my uncle for, because that's really, really what started this entire project. Was my uncle saying, you should talk to Dave and see what kind of research you could do. So thank you very much. Yes?
Have you looked at any bone morphogenic proteins or the W and T signaling pathway?
No, we haven't haven't looked at any of that. The only thing that we've really looked at is the HIPPO growth pathway. So I'd be interested to hear about other pathways that you think would be good to also look at.
Those two.
Okay. Yeah, we'll have to talk to JP and have him write it down. Other questions?